So I worked on adapting CRISPR-Cas9 to Crithidia, and that's what I fell in love with.
That would have been earlier in the history of CRISPR-Cas9 when it was like the cool thing, all the cool kids were doing.
Yeah.
Cool kids still do it.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klonowski, your host for today's episode. The people who call the shots usually get the mic, but not today.
Today, we are fortunate to have two guests join us. Kaelynn Parker is a graduate student in the Department of Cellular Biology and works in the lab of Dr. Diego Hewitt. Grace Woods Vick is a graduate student in the Department of Infectious Diseases and works in the lab of Dr. Vasant Muralidharan. Both are trainees in the Training in Tropical and Emerging Global Disease Program. Kaelynn and Grace, it's great to have you with us.
Thanks for having us.
Yeah, thank you.
I'm so happy to have the both of you on the program today. So the question that we like to ask most of our guests when they come on to the podcast is to first talk about their pathway to science. So we can kind of do that one by one, if that's okay. So Kaelynn, can we start with you?
Yeah, sure. So I had a pretty non-traditional path to science. I was homeschooled for most of my primary education all the way to the end of high school. And so when I started going to college, I was going to community college and I didn't really know what I was doing. And I took a genetics class and I just loved the class. And that's how I decided I wanted to do science. So I finished up the associate's degree and I transferred to a local university. And I still really had no idea what doing science meant. I just thought you get the degree and then you get a job. But I was really fortunate that when I was taking a cellular biology class for the core curriculum, one of my professors, Dr. Swati Agrawal, who's also an alum of the CTGD, picked me out of the class and asked me to do research in her lab. And that's how I learned about research. And I fell in love with research and with parasites. And with some not so gentle pushing from Dr. Agrawal, I applied to the PhD program here at UGA. And then
the rest is history.
Yeah, the rest is history. That's that.
Great. Grace, do you want to tell us about your pathway?
Yeah, sure. I maybe had a more traditional pathway, although I was homeschooled also. I was that kid with the frogs in her pockets growing up. So I always loved science and the natural world from a young age. When I was in high school, I actually went and did a forensic anthropology camp at Appalachian State University.
Oh, that sounds so cool.
I know.
It was amazing. And so I went to college with the intent to do forensic anthropology, which is all about bones, if you know anything about it. And while I was there, I started dabbling in more forensic molecular biology. And that's really how I got started doing molecular lab work. And from there, I got my bachelor's degree in biology, and my professors got me set up doing some forensic biology and internship in Atlanta. And from there, I got to do a post-bac fellowship at the CDC. And this really kind of built up my love for diseases. And I learned that parasites were really cool. So that brought me to UGA because of the CTGD here.
So Kaelynn, was UGA simply because your advisor was really pushing you toward her former institution?
No, not at all. It was definitely because of the parasites. I discovered that I loved parasite research and that's what I wanted to stay in. So if you want to study parasites and you're planning to go to graduate school, UGA is probably the forerunner for anyone.
And what parasite did you work in as an undergrad?
So it was not actually a human parasite. I started out working on Crithidia fasciculata, which is a non-pathogenic kinetoplastid parasite that infects mosquitoes. So my undergraduate project was to work on developing tools to utilize them as a model system for the pathogenic trypanosomes. Because it's a primarily undergraduate institute, they didn't want to work on major pathogens there. So I worked on adapting CRISPR-Cas9 to Crithidia, and that's what I fell in love with.
That would have been earlier in the history of CRISPR-Cas9 when it was like the cool thing, all the cool kids were doing.
Yeah.
Cool kids still do it.
Love that answer.
So tell us a bit about your current research. What's the main focus or main question of your current research?
My current research is on Toxoplasma gondii, which may sound familiar to even the non-parasitologists because you've probably heard that pregnant women shouldn't be around cats. And that's because cats shed the infectious form in their feces, and toxoplasma can be devastating in terms of fetal birth defects. And so our lab is really interested in understanding the fundamental cellular biology of toxoplasma, since the more we understand about how they work, the better treatments that can be developed for them. So my main project is working on understanding how their organelles communicate.
So why do organelles want to talk to each other?
Well, organelles have specific functions and materials because the compartmentalization is what allows them to be able to perform things more favorably, right? But sometimes they need to coordinate things, or they may need to share a material. So they have to be able to communicate in order to do that.
What facilitates the communication? Is it like little intracellular vesicles moving back and forth, or?
No, so there's dedicated proteins on the membranes of organelles, and under certain circumstances, they'll come together to form a contact, which is just a short distance between the organelle membranes that allows things like proteins and molecules to flow between them, or sometimes you'll have dedicated transfer proteins.
Which organelles are we talking about?
Right.
There's a lot of organelles.
There's a lot of organelles, and all organelles make contact sites. In particular, I'm interested in contact sites between the endoplasmic reticulum and the mitochondrion. And toxoplasma has phylum-specific organelle called the apicoplast. So I'm also interested in contacts between the apicoplast and the ER, and contacts between the apicoplast and the mitochondrion, which is a big mouthful.
It is. What's the approach you take to study the communication?
Very little is known about interorganellar communication in toxoplasma, and it's a very divergent eukaryotic cell. And because it has the phylum-specific organelle, we wanted to take a more unbiased approach in order to uncover some candidate proteins that are involved in membrane contact sites. So we used a method called proximity biotinylation, and basically what I did is I put a biotin ligase on the surface of each of those organelles, which I then used to create a surface proteome. And then I looked for proteins that were found on the surface of two different organelles. So like say a protein that was found on the mitochondrion and also on the apicoplast. And since we found it on both, it's likely where those organelles are close together and therefore potentially involved in a membrane contact site.
So is the idea here that if you understand what the molecules are that are important for this, you can just disrupt them for some type of targeting against the organism?
Yes, absolutely. And I also uncovered a lot of parasite-specific proteins. So like two ways that this is important. One, we're understanding how interorganellar communication works in more divergent systems, so we understand how it evolved. But two, since we're discovering parasite-specific proteins, they may be good drug targets.
Because there'd be less chance for hitting, say, the human homologue if the human homologue doesn't exist.
Exactly.
Okay. So better way to make specific drugs. And there aren't really good drugs for a lot of toxo. I know we still use things like pyrimethamine.
Yeah, pyrimethamine is the main drug that's used.
Okay, Grace, so we understand kind of the connection that Kaelynn had to UGA and why she ended up here. Can you talk about, for you, why your graduate school choice was UGA and why you wanted to continue studying parasites?
Yeah, so prior to UGA, I was at the CDC, and as a post-baccalaureate fellow there, they gave me this stipend for scientific resources, and me not knowing what to use that money on, combined with a personal love of reading, I chose to buy 20 books on diseases just across the board, polio, Ebola, malaria. And so in my free time, I was reading all of these books about diseases. Which is
kind of morbid, but...
It was cool. It was the history of public health. You learn a lot about how all these diseases impact history of humanity and how some of them are still a really big deal today. And kind of the common thread was the books I was really interested in were ones that have these really intricate life cycles. And they also tend to cause a lot of poverty. And I was really interested in the neglected tropical disease poverty cycle, where these diseases cause poverty and then you're, not able to get treatment for them. And it's just this cyclical process. And something about those really sparked my passion for science. And so when looking at graduate schools, UGA was just down the road from Atlanta. And I was like, that sounds like a really cool place to study parasites.
Did you also happen to work with someone at the CDC that collaborated with UGA?
No, actually, I was working in the Division of Sexually Transmitted Diseases. So I was working on gonorrhea, which is a bacterial disease and has a very different public health impact compared to parasitic diseases. So it's actually pretty different as far as diseases go.
Of course, as a bacteria, it doesn't have that interesting of a life cycle.
Right.
He's so biased. So I think the obvious next question is, what are the questions that you're asking now? What are you interested in?
So the lab I work in is a lab that studies Plasmodium falciparum. So this is the causative agent of malaria, which I think most people have heard of, the mosquito-borne disease. It's very globally spread and causes a lot of death and disease. And our lab is really interested in the stage that impacts humans, so the asexual blood stage where malaria is living in your bloodstream and causing cyclical fevers because they're just replicating over and over. And we're really interested in understanding how malaria replicates in the bloodstream.
And I think specifically, you guys, one of the focuses of your lab is on how the parasite gets out of the red cell. Mm-hmm. And which I find interesting, and I want you to talk about it more, because I think you pick any pathogen, and the first thing that gets researched is how does it get in?
Yep.
Which is something that I think have a good handle on now with malaria. But you're looking at kind of the tail end of that, the last part of the cycle there.
So that's how I thought my project would start, is we hypothesized the molecular machinery I was interested in was involved in the egress, and we've now really found it actually is the opposite, where it's involved in the invasion. So I think there is still a lot to be uncovered about the actual invasion of merozoites into a red blood cell, but the whole process is interesting to our lab, from the beginning to the middle, where they're exporting proteins to develop and hide from the immune system, all the way to the end where they're egressing out, and what proteins are really important in that process.
It's kind of interesting how the parasite, and talking about both Plasmodium and Toxoplasma, how they remodel the host cell. It's also kind of scary and creepy at how they remodel the host cell.
They're very good at hiding.
Yes, very good.
So Grace, what are some of the techniques and things that you use to address your research question?
Yeah, so our lab is very molecular heavy, and since my project is focused on invasion of a merozoite, this is a super fast process. And so I've really optimized the use of live video microscopy to watch in real time, taking one picture every single second, how the merozoite is able to get inside, or what proteins are involved in that process. So it's a lot of time spent at the microscope for me. So I get really beautiful videos and images, and it's cool because you can physically see what's going on. And then I think the other technique our lab uses really heavily is ultrastructure expansion microscopy. So
you're going to have to explain that.
Yeah. So part of the limitation to a lot of understanding how things work is these diseases are so tiny. And the resolution, you can't tell what's going on. And so some really amazing people came up with the process to physically expand a cell up to 4.5 times its size. And so the resolution is automatically 4.5 times better than it would be with a normal microscope. And so that gives us the ability to look more in depth at subcellular structures.
I find that absolutely brilliant, because so much has been focused on the optics. But there's limits to, based upon the wavelength of the particle you're using to image something. I think that's correct. So if you can't, with light, there's only so much you can do. But if you just turn it around and make what you're looking at bigger.
It's a long process. It takes almost a week from beginning to end. So it's a labor of love to get these beautiful images. But when you get them, it's worth it.
I love that. One of the other things that both of you share in common is that you are both trainees under the Center for Tropical and Emerging Global Diseases, or if you've heard us abbreviated here, the CTEGD. And for those of you that don't know that this is a center, it is also funded by a National Institutes of Health training grant, or a T32 as we like to call them. And those grants fund trainees in areas that, of course, they think are important. Well, number one. And number two, at areas and institutions that really have expertise and are able to train students in those areas to really, kind of go out and be great scientists in those fields. And all of those training grants have very unique things about them that make them exciting to the NIH. And one of them that the CTEGD has here is they have these capstone experiences. I know, Grace, you've already completed your capstone experience, and I know, Kaelynn, you are going to be completing your capstone experience soon. So can we talk about what those experiences are for Grace, what that was like, for Kaelynn, what you're excited about doing as part of that capstone? So Grace, why don't we start with you?
Yeah, I was so excited to go on my capstone. This is something I've looked forward to really since joining the CTEGDs, knowing that if I became a T32, I could have the opportunity to go. And so I've been dreaming about that for years leading up to it. And when the time came, I worked in collaboration with the Kyle Lab here at the CTEGD. And I was able to go to Cambodia for 6 weeks and work with the Institut Pasteur of Cambodia with Plasmodium vivax, which is a different species from what I normally work on, but is what's endemic in the Southeast Asia region. And so I was able to take that technique that I talked about earlier, ultrastructure expansion microscopy, and teach them in that lab in Phnom Penh in Cambodia. And then kind of in return, they allowed me to go out and experience some field work where they collect blood from patients that are infected with malaria. And they also do a lot of surveillance of people who may or may not be infected and are not sure if they are.
So tell us about a typical day when you're going to go out and do field work.
So I'll set the scene is that I was on a team of about 10 people. And these are people who stay out in rural Cambodia. We're actually very close to the Vietnam border. It's really far out in the middle of nowhere. And it took a 6-hour car ride from the capital to get out there. And only one person on the team spoke English. So I was very limited in how I could interact with the team. Mostly it was just miming and also sitting in the corner, waiting to be able to interact with people. And they actually have a very thorough process. So they do surveillance of small villages, and they incentivize people to come get a blood sample given by giving them things like rice and MSG and sugar in like a packet. And so you come and you're enrolled in this study, and once a month you get your blood taken, and then you get this incentive that you get to take back home. And so they actually really had a process down pat. And so part of me being there was less active and more observational, because they kind of have a system. Everyone has their table. The first person checks in the villager, and then the next person takes the questionnaire, and then the third person takes the blood, and then the fourth person gives them their incentive. And so it's this very set process. And so I actually didn't get to do as many hands-on activities as I would have preferred. And part of that is the language barrier, which was not expected. But I think the whole experience really taught me what I thought was going to happen going in is not actually what happens in field work, where things can often go very differently from expected.
That really sounds so cool. I'm picturing this first of all, the car ride.
Oh my goodness.
For
and
hours.
The roads are not what roads are in America. I would like to, I should probably start by saying that the beginning of each day, so it's a 6-hour ride out to the place we stayed in rural Cambodia. But then each morning we had to get on motorbikes and drive another hour or two. And so I, not being able to drive a motorbike by myself, was on the back of somebody else driving a motorbike.
Oh, gosh.
And road is not the word you can use to describe the paths we drove those on. It was like you were bumping up and down for 2 hours. You were numb by the time you got there.
And you really got to hand it to the people that do this day in, day out
Yeah.
For these studies. So when they take the blood, do they look at it immediately in any way, or does it go back to a lab to be checked? Or
So at the field site, they do rapid diagnostic test. of all the blood samples. I should say they do that of people who are symptomatic. Mm-hmm. The other ones get taken back and they have, I think, qPCR analysis done on them. I'm trying to remember correctly. But I know for those that are symptomatic, if they collect that blood sample, they actually put it in like a thermos at water that's been boiled, so trying to keep that blood sample warm. And they bring it back to the field site base where they have an insectary out there, which is amazing because even a lot of universities don't have insectaries.
Right.
So the fact they have this and they can feed mosquitoes and collect and get infected mosquitoes to study later on is kind of their process.
I love the opportunity to be able to see what's happening in the field. I mean, I'm sure you felt that as well when you were going out in that experience. So, Kaelynn, why don't you talk to us about your upcoming capstone?
So my upcoming capstone will be serving as a teaching assistant for a study abroad program that's held in India. It's run by Dr. Kurup, who's another professor here at the CTGD. And so for that, I will help with like the daily coordination of the undergraduates. I'll be grading their work and I'll also get to observe the same things they're observing. So this is at a veterinary hospital, so it's going to be animals coming in. I do know I will have the opportunity to see a lot of parasites that I've never seen before, like Babesia and filarial worms. So I'm really excited about that.
So an undergraduate that was doing research in my lab actually went on the last trip they had. And he was just amazed. He enjoyed it so much.
Yeah, I think thinking about this in terms of also kind of this one health structure, right? You have animals that are infected that are also kind of perpetuating the disease in these areas. It's interesting to think about that from that context and to do this kind of, it's field work almost too. I mean,
yeah, definitely. I'll be observing diseases in an endemic setting. And I think that's important for people who work on parasites to see where they're active.
And it kind of ties in with your research in that you do research on a parasite that's zoonotic. It can infect, I mean, the primary host is the cat, but it can infect any mammal.
Any nucleated cell of a warm-blooded animal is the doctrine.
That's a lot of potential.
Yeah.
It's a lot of animals.
We like to say that Toxoplasma is the most successful parasite in the world.
In that regard, yeah.
So my first question for you, because I think people are just interested in this, is what has been your biggest challenge as a graduate student?
Yeah, I think the biggest challenge would be the imposter syndrome that you face. You know, it's very much a marathon and everybody is on their own pace. When you're looking around at other grad students, even within your own lab, you're all doing wildly different things and you're all at different points in your projects. And so I think the hardest part has really been to stay focused on what I'm doing and be proud of what I'm doing and not get down in the weeds when you feel like you're not measuring up. When the reality is a lot of that is just self-fulfilling, you know.
I really like that answer, and I do want to say something that hopefully is reassuring to you, is that even faculty members can have imposter syndrome. So it's not something that's necessarily for graduate students. Sometimes we even wonder, like, can I do this? Am I good enough? Kind of thing. So I appreciate you talking about that.
So I do wonder now if you've ever thought about new folks to the lab looking at you and feeling imposter syndrome, saying, oh, I could never be as successful as Grace is.
Yeah, I know. I think when new people come in the lab or even new students in the CTGD, you want to be very transparent about, like, we're all in this together. This is hard. This is real. And this is not me versus you. Like, we're all doing our own thing and trying to make good science.
That's great. Kaelynn, I'm going to throw that same question to you.
Yeah, I mean, I agree with Grace. I think that the imposter syndrome is probably the biggest challenge. But I would say another significant challenge I've faced in the lab is I want to do all of the projects.
As PIs, we love that.
Except for when you start getting later on in the years, they're like, actually, maybe you should finish some instead of starting a new project.
Yes.
But it's a lot more fun to start something new than to finish a current project.
That is true.
It's exciting.
Mm-hmm. Yeah.
Yeah. I mean, I think that's why we got into science.
Mm-hmm.
And then we realized, oh, I guess I do have to finish these last 5 experiments, get the thing published.
Mm-hmm.
Write the paper.
Speaking of writing, how do you guys, I'll let you self-assess your writing skills. So Grace?
Well, you're going to read my dissertation in about 2 weeks, so I'm going to hope that it's pretty good. No, I think I would like to pride myself on being a very good science communicator. I think both speaking, sharing science, and then also writing science is an incredibly important skill for scientists. And I'm thankful I grew up with a mother who's actually an editor. So she was always ready with the red pen to mark down my papers in school. As a homeschooler, you understand. And so I feel like I've built a foundation of good science communication, which is something I'm very proud of, actually.
I was once told by a graduate student, or asked, to not use a red pen because it's an angry color.
David, you use a purple pen.
Sometimes.
Purple's a lot nicer of a color.
It's a little nicer.
Kaelynn, how's your writing?
I don't enjoy it very much. And I definitely think it's something I've improved a lot at while in graduate school. I'm very lucky that my PI spends a lot of time helping me edit my writing. Mm-hmm. And I do think it's improved over the course. And I will say I did just submit an F31 and I didn't get any remarks that the writing was bad.
It's amazing.
Okay.
So,
Yeah, that's always a
Yes,
plus.
excellent.
Yeah, I mean, scientific writing is a very different beast, right? And once you kind of get that formula, I think it'll become easier. So,
Grace, we might know the answer to this in your case, but what's your best experience as a graduate student so far?
The best experiences have been the opportunities I've had. So one of them being the capstone that I talked about previously. The other one being conferences that I've gotten to attend. I think they're amazing ways to network and spend time and share your research with people from other universities. And then the third kind of opportunity that's unique to my experience has been I was able to do an internship in industry here in Athens at Boehringer Ingelheim. And that was an amazing experience to broaden my parasite horizons into the veterinary health world and has also led to a job opportunity. So I think all of the extra cool things I've gotten to do have been the highlights of my PhD so far.
I love that.
Really making the most of it.
Yeah, I keep pushing Vasant, my PI. I'm going to go do this thing. And he's like, all right, well, I support you. He's incredibly supportive. So.
I love that.
Kaelynn, best experience?
Yeah, I would say as in opportunities I've gotten as a graduate student has definitely been going to conferences. So Diego's been very generous with sending me to conferences. And I've actually gotten to go to 3 international conferences, one in Mexico, one in Germany, and one in Italy. And I had never traveled internationally before graduate school, so it was a really fun experience. And I always got to vacation a little afterwards, too.
What I always like about traveling internationally is the people you meet.
Mm-hmm.
Definitely.
You know, before meeting them, they were perhaps just names of authors on a paper, and now you can say, oh, that's you.
Yeah, definitely, especially the one that I went to in Mexico. That one was actually at an all-inclusive resort in Cancun.
Oh.
And the people that hosted it were, of course, really famous PIs in both the membrane contact site world and the mitochondrial world. And because it was at an all-inclusive resort, one of the things they built into the conference schedule was beach time. And so I spent time just sitting on the beach with all these ultra famous people whose papers I've cited dozens, if not hundreds of times. And then they're just there in person on the beach.
That is amazing. I think, to have the opportunity in a very casual setting to get to know some of these people as normal people and normal scientists.
Normal people.
Normal people. We're all normal people, right? So I guess the next question is, do you have any advice for someone considering grad school? And Kaelynn, I'm going to start with you.
Yeah, I think just to try to learn as much as you can. If someone's just starting their program, if they're doing rotations, obviously you're trying to find a lab that's the right fit, but just learn as much as you can and as many techniques as you can because they might come in handy later.
And sometimes just knowing that the technique exists is helpful.
Right.
If you don't know about it, you can't plan to use it. So.
Yeah, I think that for me, something that I would tell an incoming student is to not lose the passion for what they're doing. I think it's very easy to get stuck in the middle of all the failures or the late night experiments. And for me, I am really proud to say that I've retained kind of my fun of science throughout grad school. And I think that's really important to just remember your curiosity and passion for why we're doing what we're doing and not to get bogged down in the itty bitty, stressful, tedious details.
So, Grace, we'll start with you on this one. What are your career plans? I think we might know the answer to this, but I was wrong the last time I said that. And have they changed since you started graduate school?
Yeah, I think they have changed since I started graduate school. So I'm in the unique place of being at the end of dissertation writing and getting ready to defend my dissertation in a few weeks from when this is being recorded. And I am very excited that I have a job opportunity lined up for postgraduate. So I'll be joining this pharmaceutical company, Boehringer Ingelheim, in Athens here as a postdoc, where I'll be doing molecular parasitology in the veterinary health world, so kind of looking at like ticks and fleas and kind of ectoparasites. And this is not something that I knew existed really prior to grad school. I think my capstone showed me that, fieldwork is exciting, but maybe not what I want to do. And my industry internship showed me that there's this field of animal health parasitology that I had never really been exposed to before. So that's how I chose to start pursuing that. And it'll be for the next few years. And then after that, we'll see where I go.
Kaelynn?
As of right now, I think I'd like to stay in academia. I keep saying that what I'm going to do next is a future problem, but I think the future is quickly becoming the present. So I think pretty soon I'm going to need to start narrowing down exactly what I want to work on next for a postdoc.
So our last question that we ask our guests are, what do you like to do in your free time when you're not thinking about parasites?
I think I already hinted at this. I'm a really big reader. So I average over 100 books a year read, even during grad school. So I spend a significant amount of time just reading for fun, in all genres, really. I'm all across the board.
I also really like to read. I don't read quite as many books as Grace does.
She's a little bit of an overachiever there.
I also really love bird watching. So one of the classes I took in undergrad was ornithology, which was basically guided bird watching, and it really sparked a big hobby for me. So I do a lot of bird watching now.
It's interesting how classes we took as an undergrad can really influence us. We all remember that one class, perhaps, like Kaelynn, yours that got you interested in genetics. For me, it was molecular biology.
Shocker for me, it was immunology.
Really? Would not have guessed.
Kaelynn and I both like bird watching, and so we'll send each other bird alerts when we see exciting birds outside the third floor covered windows. We'll go take a break outside and watch the birds.
Okay, well, our guests today are Kaelynn Parker and Grace Woods, who are graduate students in the departments of cellular biology and the departments of infectious disease, respectively. Kaelynn and Grace, thank you so much for being with us.
Thanks for having us.
Yeah, thank you. This has been really fun.
So links to additional information about Grace and Kaelynn's research can be found on our Instagram account, which is at PPP Podcast UGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season and this season. Please rate the podcast, and if you have any questions or suggestions for future podcast episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemitz, for research, editing, scheduling, and keeping us on track.